Hello and welcome back to Simply Biology. In today's video we're going to be looking at apoptosis. We're going to be looking at what it is, comparing it to a process of necrosis, and also looking at why it is important and the actual process of it. So without further ado, let's get started. So apoptosis is the process of controlled and programmed cell death. Cells can be destroyed by two processes, either apoptosis or a process known as necrosis. Like we've just said, apoptosis is the internal controlled programmed cell death, whereas necrosis occurs by the rupture of the cell membrane and the release of hydrolytic enzymes, either caused by an infection or a trauma. So obviously this is very bad and can damage the cell, whereas this is the internal controlled mechanism for the regulated cell death. So, as I just said, it's a regulated process, and it's regulated by hormones and proteins known as cytokines. Apoptosis can be halted and initiated by P53, which is a tumour suppressor gene, which we'll learn more about in a later video on cancer. Important for the formation and the connection between neurons in the brain, which are just nerve cells. Neurons are also known as nerve cells, so it helps to form neurons in the brain. It can be used for the destruction of harmful immune cells during the development of the immune system, and it's also very important for the production of distinctive digits such as hands and feet. So when you develop in the womb, what happens is all the cells in between your fingers. And what happens is these cells die by a process of apoptosis in order to produce the distinctive digits that we can use for including our hands and feet. So we're now going to look at the actual process of apoptosis. So first of all, the cell shrinks and the nucleus condenses. And the condense of the nucleus is what's known as pinosis. The enzymes then break down the cytoskeleton. So obviously this is the internal control and transport system within the cell. The next stage is the cell membrane alters and it forms bulges known as blebs. The nucleus then fragments and begins to break down in a process known as karyohexis. The cell is then broken into vesicles, so all the cell fragments are then broken down and separated into vesicles, which are just fluid containing sacs, and these are known as apoptotic bodies. Phosphatidylserine is a phospholipid that is usually found on the inside of vesicles, but it is present on the outside of these apoptotic bodies. What happens is this phospholipid binds and activates immune cells known as macrophages. These macrophages are just differentiated monocytes which have left the bloodstream. And what happens is once these are activated, they engulf and digest the cell fragments, and therefore the cell is died and it is just being destroyed in a controlled manner. So I hope that's cleared up the process of apoptosis, and I hope you've learned something very useful from today's video. Just remember that apoptosis is the controlled, regulated and programmed cell death, whereas necrosis is the traumatic event which leads to the release of hydrolytic enzymes, and that can be very, very damaging for the cell. Thank you for watching and goodbye.